Welcome also from my side. I have the honor to welcome today's speaker, Dr. Sabrina Borchert. He is currently a postdoctoral fellow at the Institute of Pathology of the University Hospital Essen in Germany. And her scientific objective is to understand the longitudinal dynamics of translational cancer research, lung cancer, and rare malignancies like sarcoma or malignant pleural mesothelioma. Today, she will focus on the work she and her team did on sarcoma, and she will present the work done on the basis of the HTG Transcriptome Panel. So thank you so much, Sabrina, for being here today and for being our speaker.
Yeah, thank you very much for your invitation. I'm very pleased to present to you today our published study about the comparison of synovial sarcoma with spindle cell sarcoma with the help of the HTG Transcriptome Panel, shortly named HTP Panel. So this is my disclosure information. And the background of the study is that synovial sarcomas represent a group of soft tissue sarcomas affecting patients at a wide age range. Thus, it could include adolescents and young adults. And mostly, synovial sarcoma is clinically aggressive. Many patients suffer from local relapse or distant metastasis, and even after more than 20 years, late progression can occur. And synovial sarcoma is a paradigm for gene fusion-driven cancer because it is characterized by an in-frame fusion of the SS18 gene with either SSX1, SSX2, or in rare cases, SSX4. So this is a little polling question, and I would be happy if you could take a moment to answer it. So which of the following best describes your role? So A, healthcare provider, molecular pathologist, research scientist, or bioinformatics or computational biologist, or E, other or non-scientific role. Yeah. Should I go ahead?
Yes, you may continue. Thank you.
Thank you. So spindle cell sarcomas are the morphologically similar to synovial sarcomas, but they behave clinically different and thus are the so-called morphologic mimickers. And these do not exhibit the gene fusion of the chromosome X and 18. And the aim of our study was to compare these two entities, but the main part of the study was to test the robustness of the HTG transcriptome panel with the results of already established methods in the routinely diagnostic, like immunohistochemistry, fluorescence in situ hybridization, or RNA-targeted sequencing. And this is the polling question number 2, so you are welcome to take a moment to answer it. So have you been involved in studies using FFPE samples to produce bulk gene expression profiles? If so, do you recall which technology was used? Yeah, I will go ahead. So the HTG Transcriptome Panel consists of about 20,000 targets and includes various types of controls. And we analyzed samples that were formalin fixed and paraffin embedded, so shortly named FFPE material. And we used 1 to 2 sections per block, depending on the tissue area of interest. And yes, the library was prepared by using the HTG device and a thermocycler for tagging of the sequencing adapters for the subsequent sequencing on an Illumina NextSeq. And as you may know, the advantage of this technology is that RNA isolation is not needed, hence it is performed by the HTG device. So this is a simplified workflow with less hands-on time and low risk of RNA contamination. Yeah, and as I said, the panel consists of 20,000 probes, which hybridize to the target RNA. These are the so-called wing probes. And after hybridization, the target RNA is eliminated and in step 4, the adapters and tags are added, generating the libraries, which are then ready for the sequencing step on the Illumina NextSeq. And we analyzed the results by using the HTG HSEQ Reveal software. It is a powerful tool to perform data quality checks, to normalize the data, and to perform correlation plots on replicates. And also it supports differential expression analysis and signature development and validation by generating volcano plots, heat maps, PCA, principal component analysis cluster analysis, and box plots of the most differentially expressed genes. So first of all, we have to check the quality of our HTG run. So there are 4 QC metrics. So the QC0 gives us information if we have sufficient RNA in our samples. And the QC1 gives information about the read depth of our sequencing run. The QC2 gives a failure if we have a too high background signal of our samples. And the QC3 gives a failure if we have an incomplete digestion of genomic DNA by the DNAs. And in our plots, we can see we have sufficient RNA in our samples, and we have sufficient read depth, and we also have not a too high background signal in our samples. So a few samples show a higher background, but they are still below the threshold line. And in our QC3 plot, we can see there are 2 samples failed. They are very near to the threshold line, but still they failed. However, this plot is in the previous version of the Reveal platform as we have published it. So we published it in this form, but Reveal is steadily optimized so that the current version of the Reveal platform would show that each sample has passed the QC. But I will show you this plot because it is published in this form. And the current version is shown in this plot, and you can see that each sample has passed the QC, so we have a complete digestion of genomic DNA. And this is an H&E staining, so hematoxylin and eosine, of synovial sarcoma and spindle cell sarcoma to show that they have a really close morphology. But our principal component analysis shows that the gene expression pattern of both entities is clearly different. So the yellow dots indicate synovial sarcoma and the green dots indicate spindle cell sarcoma samples. And also this volcano plot underlines the differences by showing about 6,000 genes that are significantly differentially expressed. And these are the top 10 upregulated genes in synovial sarcomas and the top 10 upregulated genes in spindle cell sarcomas. And it's very interesting and also important that the TLE1 gene is present in the top 10 upregulated genes in synovial sarcoma, as it is an already established marker for in diagnostic routine. So you can see also here So the Reveal platform could also show bar plots of the gene expression of each gene to visualize the different levels of expression. So the two bars on the left side indicate the expression of TLE1 in spindle cell sarcomas, while the other two bars show higher expression of TLE1 in the synovial sarcoma samples. And this is an example for the immunohistochemistry of TLE1 in spindle cell sarcoma on the left side. So it's clearly negative for TLE1, and in synovial sarcoma, the staining is clearly positive, so nuclear and cytoplasmic. And yeah, it's interesting that the protein expression correlates with the RNA expression we could detect with the help of the HTP panel. And also the expression of SSX1 and SSX2 in the samples with the respective gene fusion is visualized by this plot. So SS18 is evenly expressed in synovial sarcoma samples, while the patients exhibiting an SS18 SSX1 fusion do not show expression of the SSX2, and the other way around. And this is the last polling question, so you are welcome to take a moment to answer this question. If you paid attention, you can answer it. So approximately how many genes can be profiled using the HTG Transcriptome Panel? So A, 2,000, B, 10,000, or D, 20,000. Thank you very much. And so we also compared this technique with other established techniques from this table, including the Lexogen QuantSec, QIAGEN QIAsec, BioSpider TempoSec, and so on. And this is a comparison table by Turnbull et al. So this is here. And these techniques were compared in terms of biochemistry, sample throughput, number of primers, mapped gene IDs, and read depth and input of FFPE sample RNA, costs per sample, and so on. And to supplement this table with info about the HTP panel performed in the study, we listed these parameters also in this table. And the HTP panel has a moderate sample throughput with 24 samples per 3 days. When performing with the 24 sample kit, it is also possible with 96 samples. And the number of targets of about 20,000 genes targets, as well as the sequencing depth, is close to comparable methods such as BioSpider TempoSec. But its sensitivity and success rate of FFPE samples is the highest compared to others, considering the low RNA input that is needed for HTP. And in addition, the FFPE blocks used in the study were stored up to 5 years, and HTG molecular itself reports robust results for FFPE material older than 10 years. So I come to our conclusion of this feasibility study. So it is a very feasible method for gene expression analysis in a large scale, and it revealed robust results. And it is a robust method for the search of biomarkers, especially in limited tumor material. So we are talking about FFPE material, which is the best available material, but it's very challenging to measure RNA in these fixative material. But even analysis of biopsy material was possible with the HTP panel. And yeah, we could also prove the already known overexpression of TLE1 in synovial sarcomas, which underlines the reliability of this panel. And now I would like to thank HTG for giving us the possibility to test our samples and to present our study in this webinar. And thank you very much for your attention. And if you have questions, here's my email address also. And I'm glad to answer questions now.
Thank you so much, Dr. Borchert, for this wonderful presentation and for sharing with the audience the insights, the valuable insights you gained in identifying differentially expressed genes between synovial sarcoma and morphologic mimickers. So the audience is now invited to use the chat function to type in the questions we would like to have answered. And I can see there's already one question. In the presentation, it was mentioned that one of the attributes of the assay that was used was low sample input. Can you confirm the minimum amount of sample that was used, and how does that compare to other approaches you have used?
Yes, as I said, we could also get robust results from biopsy material. And yeah, with fluorescence in situ hybridization, it's also possible to get a robust result, I think. But RNA targeted sequencing would be challenging with biopsy material, if that answers the question.
Okay, thank you. The next question is, what are the next steps in TLE1 biomarker-related research? Are there any ongoing clinical trials focused on this biomarker?
I don't think so, because it's a really discussed biomarker, as some papers also report that TLE1 overexpression is not only one marker for synovial sarcoma, but for other sarcomas. However, in this study, we also could, with the comparison between synovial sarcomas and the spindle cell sarcomas, we could clearly show that TLE1 was overexpressed in comparison to the spindle cell sarcoma. So in this type of sarcoma, we could prove it. But there are so many other entities of sarcomas. So that would be interesting. So it would be interesting to make further studies on it. But it's a very, very rare cancer. So it would be challenging. But pathologists still say that TLE1 is a good marker for synovial sarcoma. They still think this sorry?
They still think TLE1 is a good marker despite the limitations of immunohistochemistry.
Yeah, thank you.
Okay, I understood. And so the other part of the question was, are you planning to validate that biomarker further? Or the other, what I add is, do you plan then also to validate the differential expressed gene signature further, so that this could potentially even become applicable in routine?
Yeah, this is really planned, so for sure.
And so, do you see this gene expression classifier being applied in clinical routine to better differentiate synovial sarcoma versus the mimickers?
Yeah, maybe that's a good option.
Okay, thank you. So I'm looking at the chat. For the time being, we don't have any further questions. You still have time to enter any questions if you want. But if there are no further questions, then I thank you so much, Dr. Borchert, for this great presentation and the great work that you did. Wish you best of luck for the further validation and the next steps to bring that closer to the patient, and congratulate you to the great work that you're doing.
Yeah, thank you very much.
Thank you. And thank you everyone for joining. Another great webinar, and thank you so much for your attendance. If you have follow-up questions, also Sabrina shared the email address. We can reach out to her directly if there are any questions coming up in the retrospective. Thank you all. Wish you a great rest of the day.